Okay, today we want to talk about Chapter 16, the chapter on DNA, the genetic material, which we'll get back to these guys in a moment, Francis Crick and James Watson. We'll see their role in a few moments. But first, in the early 1900s, DNA was known of, and it was known that it existed in the cells, and it must have some kind of job there because it was in the cells, it was in the nucleuses of just about all cells, and cells like to make copies of it. So, but people weren't quite sure what it did. But there were some early experiments that gave insights into this, including the Griffith experiment. And Griffith was studying these bacteria, some that were pathogenic and some that were not. And you can see the pathogenic one, of course, kills the mouse, and the non-pathogenic one does not. When you heat up the pathogenic ones, the mice are just fine because they've been killed and activated, I guess. But if we mix the healthy non-pathogenic with the killed pathogenic type, we let them coexist for a little while, and then we inject them into the mice die. And then if you extract fluids from the mouse, you indeed find the living pathogenic strain. Essentially, Griffith had discovered what we call transformation, or bacterial transformation. Okay, so now there were questions about, okay, what is it that allows these non-pathogenic bacteria to be transformed? And an experiment I don't have pictured here, but it was Oswald Avery in the middle of the 20th century, Avery, who did some experiments where what he did was he recreated the transformation experiment, but this time he took those bacteria that had been killed and separated out different components. Particularly, he was separating out proteins from the DNA. And so he's curious, okay, which of these two is the transforming agent? And when he exposed the non-pathogenic bacteria to proteins, nothing happened. They stayed that way. But when he exposed them to DNA from the pathogenic bacteria, the non-pathogenic type were transformed. So he essentially demonstrated that DNA was the transforming agent in these bacteria. So that's pretty important. All right, next, the Hershey-Chase experiment. Now here in this one, we're making use of a phage, which is a virus that infects bacteria. So we got the phage and we have bacteria. Phages, like all viruses, consist of an outer covering called the capsid, which is made of protein, and then they have a nucleic acid on the inside, and some are RNA viruses and some are DNA viruses. So they were curious, okay, what is it in the viruses that allow them to take over the bacteria, that allow them to take over the host cell? So they basically cultured these viruses in the presence, one batch in the presence of radioactive sulfur and one batch in the presence of radioactive phosphorus. Now, sulfur is found in proteins, but not nucleic acids, and phosphorus is found in nucleic acids, but not proteins. All right, so they allowed the radioactively labeled phages to do their thing with the bacteria for a while. Then they put the mixture in a blender to separate the different components, basically the viruses from the bacteria. Centrifuge the liquid. The bacteria, being larger, settle on the bottom and form a pellet. The viruses stay in the liquid part. And what happens? Well, when the sulfur had been labeled, the radiation is up in the liquid with the virus particles. When the phosphorus in the phages is labeled, where does the radiation end up? It ends up inside the bacterium. So it appears that the viruses are using their nucleic acid, DNA in this case, to take over their hosts. So again, DNA having a very important role there. And here's a electron microscope picture of some of these phages, these robotic looking like viruses infecting their host there. So these experiments are demonstrating the importance of DNA. And a bit later, in the 50s, we start getting some folks working on, okay, we know generally what DNA is made of, but what's the structure like? How are all these different components arranged? And so it was the work of Watson and Crick, and also, most importantly, of Rosalind Franklin, who was using X-ray crystallography to essentially, quote, take pictures of the DNA. And so by shooting these X-rays at the crystalline form of these molecules, the X-rays bounce off and you have film behind the molecule, and essentially where the X-rays are hitting is where the film gets exposed. And so when you do this with different molecules, you get different kinds of patterns. And here with her photo 51, this X-ray pattern, this X-shaped pattern is apparently, if you're an expert on X-ray crystallography, is characteristic of a helical-shaped molecule. And so Watson and Crick were able to make use of Franklin's information, her data, without her knowing about it. And building their models, they essentially figured out the correct shape of DNA. That it's a double helix, the two strands consisting of, on the sides, the sugar phosphate backbone of the molecule, if you will, on either side, and the nitrogen bases in the middle. And by using also some other information that was available, they didn't have to take this, from Erwin Chargaff, he came up with Chargaff's rule that the amount of adenine is equal to the amount of thymine and the amount of guanine is equal to the amount of cytosine. They, again, using that information as well, were able to figure out the shape of the molecule, with the adenines bonded to the thymine in the middle and the cytosines bonded to the guanine. All right, so the nitrogen bases are in the middle with A bonded to T and C bonded to G. And one thing I want you to notice is, and you may remember this from freshman year, is that in addition to being complementary, the DNA molecule, the two sides basically run in opposite directions. The sugar, in particular, has, of course, an organic molecule with carbons in it, and so there's a carbon here and here and here and here and up here, these 5 carbons, and so those are numbered. And so this is the number 1 carbon to 2, 3, 4, and 5. And you notice basically this is sort of the 3 side of the molecule, and this is the 5 side. And the sugar is oriented in the opposite direction on the other side. So we label those different sides of the molecule, the different sides of the strands, as the 3' end and the 5' end, and again in the opposite direction on the other side. We'll see how that plays a role when it comes to DNA replication. Now, apparently the X-ray diffraction pictures give you lots of information about the shape and the dimensions of the molecule, and so when Watson and Crick started playing with the nitrogen bases, if you put 2 of the purines together, and again the purines are adenine and guanine, and the pyrimidines are cytosine and thymine. So if you put 2 purines together, it's too wide. If you put 2 cytosines, it's too narrow. But if you put a purine to a pyrimidine together, it's just the right distance. And the reason the A and T get together is because they like to form 2 bonds, whereas the cytosine and guanine like to form 3 bonds. So you won't get the thymine with the guanine or the cytosine with the adenine. All right, so let's start on section 2. See how far we get. Okay, so Watson and Crick mentioned in their paper about this potential mechanism for the replication of DNA because of the complementary nature of the bases. If you know one side of the molecule, you automatically know the other side of the molecule. And so they postulated that if you just unzip it, you basically have the template to build up new complementary strands of DNA. And so that's what they had proposed or hypothesized, and that fit what is known as the semi-conservative model of DNA replication. And that is when you make a new strand or a new copy of DNA, essentially one side of it is old DNA, the dark blue here, and one side is new DNA. But others had thought, well, maybe when you make two copies, one of them just retains all the old stuff and the other copy gets all the new stuff, the conservative model, or it's relatively randomly dispersed, the dispersive model. Well, it was these folks, Messelson and Stahl, who did some experiments that demonstrated that it was the semi-conservative model, because what they're using is a radioactive label, a heavy isotope of nitrogen here. And so they basically cultured some bacteria with heavy nitrogen for a while, and then transferred the culture, and then they would have been going through binary fission and copying their DNA, etc. Then they transferred them to the regular lighter isotope of nitrogen and let them do their thing for a while. So you're going to have some of the DNA in this sample that has the heavy isotope and some that has incorporated the lighter isotope in later stages of replication and such. So let's see what happens. So now, all right, so if you have the original DNA that's just the heavy isotope, you're going to have a really heavy band when you centrifuge this. That's going to be the bacteria essentially that have incorporated the heavy isotope. Then you're going to have some of those bacteria that have the lighter isotope. Well, that's not what they found after the first replication. That is, they just found a single band. So it was somewhere in between the heavy and the light. Okay, so now they moved to the next step and said, okay, well, after the second replication, what should happen now? If it's the dispersive model, that heavy isotope should just get equally dispersed amongst all those new copies, and they should all be about the same mass, and you'll have a single band when you centrifuge it. But what they found was, when they centrifuged it with the batch that had gone through a second replication, they had a band that was in the same place as in the first replication, but then a very much lighter band. So essentially, after the first replication, when we went to the second replication, you had these two pieces of DNA essentially that still retain that heavy isotope. But think about it, when you unzip these guys and make new DNA, there's going to be one of those copies that is exclusively new copies, if you will. That is, they've incorporated just the lighter isotope. And so that was confirmation of this semi-conservative nature. That is, when you replicate DNA, you have sort of one old piece and a new piece, one old piece and a new piece. Okay, Messelson and Stahl. Okay, we'll stop right there and get to replication in the next video.